And good morning, everybody. I think you saw our short title. It's a small animal model of COVID-19 infection. And why this is important, I'll give the justification for this, but basically it's that mice and rats, the workhorses, so to speak, from the laboratory, cannot be infected with this virus unless there's a transgenic form of them expressing the human form. So that leaves a more limited set of possibilities. Non-human primates obviously would be ideal, but they are very expensive and many challenges to work with that. Hamsters, Syrian golden hamsters can get infected, and I think Chinese hamsters as well. And this could be a suitable model, but the hamsters are supposedly outbred, but the hamsters that we deal with have all been derived from a brother and sister captured near Aleppo, Syria in the 1930s. So they're effectively inbred animals. And the interest of our group, which is Don Forthall, as well as Ilhelm, Tony Long, Mike Buchmeyer, is to make use of this unique genetic structure, population structure of the animal that we're working with, the white-footed deer mouse, Paramyscus Copis, that makes it very suitable to, in terms of its genetic diversity, to a human population. But unlike a human population, most human populations, Paramyscus leucopus, the colony that we deal with was started with 38 founders in the mid-1980s and has been a closed colony since then. So it's like an island population now. And so there's a great deal of, there still retains much of the genetic diversity of the species, but it has a lot of linkage disequilibrium, which means that GWAS can be done, sufficiently powered GWAS can be carried out with, say, 500 animals instead of several thousand, which would have to be done with humans. So that's the background on the animal itself. This is the reservoir for Lyme disease, several other human diseases in the United States, sister taxon, Paramyscus maniculatus is the reservoir for hantavirus. So it functions very well as a reservoir for human diseases, and it's analogous to bats in that sense. So why that's the background. I looked at the sequence for the AC2, which serves as the receptor for the virus, and saw that it was fit very well with the predictions of what would be a good receptor for the virus. Tony Long and I had sequenced the genome of this animal. We had RNA-seq on many tissues, and we showed that the ACE2 homolog is expressed in the lung of these animals as well as other tissues. We've identified 42 million SNPs to date in this population, so it makes it very suitable for doing GWAS studies. So we don't have any more preliminary data than that. The plan is to infect these animals with the virus. It'll be done in BSL-3. We should be able to do it in about 2 weeks. The virus is here. We're working with Ilhelm. We're going to try it in lung tissue, excised lung tissue to begin with, and then use intranasal administration of the virus, and then within a week, we should know if there's been replication. My prediction is that they will get infected. They're not going to get terribly sick or die from it. They'll tolerate it, but there'll be a range of responses which would make it suitable for a GWAS approach.
Okay, thank you very much. I guess we'll open for questions now. So we have 5 minutes for questions, maybe a little less to make sure we stay on schedule. I should just ask people, if you signal in the chat box that you have a question, we can proceed really efficiently through these. But for now, I'll open the floor if anyone has a question.
I have a question for Alan. Hi, Alan.
Okay, great. You're supposed to know all about this.
I know. I was just wondering if you looked at the expression of the known co-receptors as well in your RNA-seq data.
Which receptors? I didn't hear that.
The co-receptors like the TMPR, SS2, and those other co-receptors that like more and more papers are coming out to say.
Okay, no, that's a good idea. Well,
yeah,
we can do that. Right. Yeah. I'll bring that up. Right, thank you. So which are the ones that you would focus on, or what do you think is most important?
I'll send you a list, but one of the most important ones so far is the TMPR SS2.
Yes, okay, yeah, I know about that one. All right, thank you.
Question, Alan. This is Eric Perlman. I'm over here.
Yeah.
Really exciting model that you have there. I'm just wondering, how do you see this compared with, you know, we're planning with Tom Lane and Bert and others and Ilham to look at the human ACE2 receptor transgenic mice, and Tom is in the process of getting these mice. How do you see these outbred mice in terms of us being able to do the sort of standard experiments where you infect them, you look for cytokine responses, you look in the tissues to see what kind of level of disease? Do you think there'd be just tremendous variability compared with the transgenic mice that are on a V6 background?
Yes, well, that's the whole point, that they're variable, and one can do GWAS with them. You can't do that with transgenic mice. You can't do that with ferrets. Cats would be a challenge as well. So that's the whole point. You know, we don't have antibodies to all the cytokines. We have RT-qPCR for several cytokines now, and we can do RNA-seq to look at the sorts of profiles that you're talking about. So, yes, we don't see it as a competition to the transgenic mice, as complementary to them, and to serve as a model to try to understand, what's going on in bats. I think, at some point we've got to try to understand what's going on in these reservoirs to make them so suitable for harboring all these viruses and bacteria and things like that. And this would be a start.
Actually, this is Tali. This is really, really exciting, because one of the questions that I think we began to talk about, Alan, is what makes some people more susceptible to others? Clearly, obesity and what have you seem to, once you have an infection, as well as age, makes the infection worse. And one of the obvious questions that come to my mind is something, what about stress? Because it is the most potent suppressor of the immune system that we know of. So having an animal model that represents a diverse genetic background is fantastic, not only for genetic studies, but also, if you will, for acquired or maybe gene-driven issues and aspects that modify the progression of the disease. So I'm really thrilled that you're doing that.
Yes, thank you. Yes, so yes, and what Tali was saying about the sort of the host response and variability in that, Tony's already identified some SNPs in the AC2 molecule already that are common variants in the wild population. So that suggests that we're going to find further variation in other parts of the genome.
Alan, it's Mike. I just think it's important to remember that these are not mice. These are leucopas. And just to differentiate, what do you think the differences are that might prove to be important later on in terms of interpreting this data from the mouse window?
No, Mike's right. And I should have mentioned that they're more closely related to hamsters and voles. They're in a different group of rodents than murids. So again, if I go on the limb here, I mean, we found in some other studies, we're working with Ali Mortazavi and a graduate student, Anna Milovich, is that these animals tend to have more of a macrophage 2 type response to infection and LPS. So I think they're more on the err on the side of anti-inflammatory responses than the murines would.
And that's exactly, if you're considering that the innate response, that's exactly where bats seem to fall short in the response to pathogens. You know, bats really tolerate pathogens much more than we do, and they don't overreact. So they're like a 10-year-old who's infected with COVID, you know, who doesn't show an extravagant response and is able to carry the virus for a period of time. Now, we don't know how long bats carry. It could be a long time, it could be a short time. We really don't have a handle on that, but that's important. We see,
Right.
by the way, the same kind of species restriction with the New World arenaviruses. And they pick different chrysotine rodents as their natural host. And it may, in fact, even serve as an environmental factor in differentiating the populations of these rodents. You don't find overlapping populations in the same rodent of virus carriers.